So the most enigmatic aspect of cancer is the progression of the tumor. Why do some tumors stay benign and never travel to other places, and others disseminate throughout the body? Here in the case of a breast cancer that originates in the breast and the cells spread to the proximal lymph nodes and end up forming colonies predominantly in the bones and in the brain, and that in the end kills the patient. This is the most undertreated and least understood aspect of cancer. If you allow me, rather than giving you a data-heavy talk on one paper, I would like to give you a little bit of a summary of several years of research that we have done into cancer, some of the key questions that we've tried to answer, and the results that we've gotten from them. First one is, does metastasis have a genetic basis? There was a big debate in the 1990s. Is metastasis genetically controlled or is it epigenetic? 1990s were also the year when the first bona fide metastasis gene was identified in CD44. It was actually a CD44 splice variant that was identified in pancreatic cancers, and it became a very hot topic. So, increasing number of publications and increasing number of functions were attributed to the surface molecule CD44, homing receptor on lymphocytes. And so at some point, it became very unclear what the real roles were of CD44 because the literature was so broad. So we tried to test the role of CD44 in the closest system to human cancer that we could come up with. And those were mice with a spontaneous mutation in P53, the P53-TM1 mice. They develop endogenous tumors, so not injected, not transduced. So these are endogenous tumors. And they include, beside a number of lymphomas, very aggressive, very dedifferentiated osteosarcomas. The osteosarcomas in these mice metastasize. They form lung metastases, and you see very nicely here the osteoid formation of a pulmonary metastasis. And they form metastases in the liver, and you see the destruction of the liver tissue by the metastasis. Now, these mice die from their primary tumors because the primary tumors become so big. So we asked, when the mice develop these cancers, do they have the same tumor spectrum? And the answer was yes. Do they have the same lifespan? And the answer was yes, because they die from their primary cancers. So what about the dissemination of these tumors? What about the metastasis? As I said, each and every one of the P53 mutant CD44 wild-type mice had multiple metastases in the livers and in the lungs, up to 13 lungs found in step sections. Right, a section through the lungs. In mice, you have to really look for the metastases. In the P53 CD44 knockout mouse, there was one solitary metastasis and one lonesome mouse. So you knock out CD44 and metastasis is almost entirely abrogated. And because we were working on the ligand for CD44 called osteopontin, osteopontin being a cytokine, it's used in tissue remodeling, but pathologically it's upregulated in cancers. And we formulated a model which we believed explained how things work. Osteopontin is cleaved in the middle of the molecule by thrombin, and the C-terminal end binds to CD44 and induces chemotaxis, whereas the N-terminal part binds to integrin alpha V beta 3 and induces cell crawling on a solid surface called haptotaxis. So we envision a two-step process here where the C-terminal end attracts the tumor cell in circulation to the organ of destination, and once it's there, osteopontin N-terminal part via alpha V beta 3 makes the cells crawl on the endothelium. At some point, the cells reach the highest concentration of osteopontin, they have nowhere to go. Cells always migrate upward, a gradient, and never downward. So when the gradient, the highest level of the osteopontin concentration is reached, the cells spread, the spreading induces metalloproteinase secretion, and that's how we envision the cells going into the target tissue. So we thought that we had learned from those studies that osteopontin and variant CD44 interact, and that interaction was essential for metastasis by several types of cancer. And actually, CD44 is very commonly expressed and aberrantly spliced on various solid tumors, and osteopontin is responsible for metastasis of about 30 different cancers. So that led us to the next question: how are these genes actually dysregulated in cancer? As I mentioned initially, there was a big debate in the 1990s. Is metastasis genetically controlled or is it epigenetic? We didn't have mutations in the coping regions of these metastasis genes, and that's what caused that debate. So how are they deregulated if they are not mutated? But first of all, just to confirm that osteopontin is a cancer progression-associated gene, this was a meta-analysis done from 248 papers, covering about 13,000 patients, and it does confirm very nicely that osteopontin correlates with progression of the cancer as measured by grade, or as measured by in the T and M staging, with the stage of the cancer, with T and N. And actually the number of studies for M stage for the metastasis was a little bit low, so we got a p-value that was borderline compared to the others. But we believe that there is significance. So osteopontin is associated with tumor grade and tumor stage. Needless to say, and unsurprisingly so, it is also associated with the survival of a number of cancers, a number of very common cancers, lung, breast, prostate, head and neck, and liver. You see the correlation being very strong, and you also see that the p-values are very low. So very convincing, very trustworthy association here between tumor progression and osteopontin. But as I said, osteopontin is essential for tissue remodeling. So what happens in cancer? What makes osteopontin different in cancer from physiology? Well, we had come up with the idea that metastasis genes in cancer are not mutated, they are apparently expressed or spliced. Osteopontin does actually have splice variants. You see the full-length form on the top, and you see two splice variants lacking either exon 4 or exon 5 called osteopontin B and osteopontin C. Right? So these two forms, thus far, we have only found in transformed cells. We have never seen osteopontin B or osteopontin C in a non-transformed cell. And when we look in breast cancer, we see that osteopontin A, the full-length form, the unspliced form, is actually very variably expressed in normal tissue, in tumor surrounding tissue, and in breast tumors. Not surprising, because osteopontin actually is induced by estrogen. Osteopontin B, the intermediate form, is absent, and actually we know that it's degraded in the proteasome. The RNA is made, but the protein is unstable in the breast. Osteopontin C is here in the cancer. It is there in the tumor surrounding tissue, but when we look in normals, it's totally absent. Right, so now we have a marker that is not only quantitatively overexpressed in cancers, that is qualitatively different between the normal tissue and the cancer. When we do immunohistochemistry, we see what we had seen before, that there is an increase with grade. And this is now osteopontin C specific. This is not total osteopontin. And we see that the normal breast is totally free of osteopontin C. Right? And we do this quantitatively with the pathology scores. We see this correlation very nicely, and we see the total absence from the normal breast. And that actually distinguishes osteopontin from the three classical breast cancer markers, HER2, ER, and PR, that show absolutely no correlation with tumor grade. Right? So we learned now from these studies that in cancer, metastases genes are aberrantly expressed or spliced. They are not typically mutated. When you have a mutation in metastasis gene, it's in the regulatory region. It's not in the coding region. So what are the mechanisms? Right in the 1990s, late 1990s, when we got into the field, the homing, the directed migration, was believed to be the major aspect to explain metastasis. Today we know that actually tumors, malignant tumors, shed cells on a daily basis into the circulation. 99.99% of these cells do not survive. There's only the 0.01% of the surviving cells that have the potential to cause clinical metastasis formation. So the survival of the tumor cell in the circulation is actually the main limiting factor in metastasis. So we looked at what osteopontin does to the survival of these cells. And one way to measure this is you put tumor cells into a semi-solid medium, into soft agar. Right? You have a high concentration soft agar that you have on the bottom so that the cells don't attach to the plastic of the plate, and on the top you have a lower percentage soft agar within which the cells are. If these cells have no anchorage independence, then they die. And you know, when you take an epithelial cell that's very stringently dependent on its surroundings, right? The substratum, the adjacent cells, the shape of the cell, all determine the survival of the epithelial cell. An epithelial cell that loses that contact undergoes a form of programmed cell death called anoikis. So the anoikis is something that all of our non-transformed cells are subject to, whereas the tumor cells may be resistant. A fraction of the tumor cells is resistant, and soft agar is a pretty good readout for that. Now, we transfect these tumorigenic non-invasive cells with osteopontin A, and we do see some increase in the size of the colonies. You see how tiny the colonies are here in the untransfected cells? They do become a little bit bigger in the transfected cells. So osteopontin A does increase the size of the colonies in soft agar, but the transfection of osteopontin C is tremendously more powerful. You see these huge colonies, and you see how they stretch out protrusions into the surrounding medium. We think that this is a matter of very rapid growth of osteopontin C. And when you actually measure this and add a neutralizing antibody, an anti-osteopontin antibody, you see that you can reduce the clone size, the colony size in soft agar, indicating that osteopontin has to be secreted. So it's an autocrine mechanism. Osteopontin is produced, it's secreted, binds to a receptor, and causes survival. And that's why we can inhibit that with a specific antibody that is given extracellularly. And we were able to extract the cells from soft agar. We were able to take out the cells, extract the RNA, and do a microarray to answer the question, what are some of the molecular mechanisms? What are some of the gene expression mechanisms that lead to anchorage independence? And somewhat to our surprise, we found that the biggest category of genes were oxidoreductases. Some of them were electron transporters, but a lot of the electron transporters also function as oxidoreductases. So we believe that the oxidative metabolism played a key role in allowing the cells to survive under anchorage-independent conditions. And again, we tried to make sense of the data in terms of building a model. Remember, we came now from the directed migration to a change in paradigm where the anti-anoikis is the predominant factor and the migration is secondary to this. So we believe that the tumor cell, once released from the substratum, actually is able to survive, which a normal untransformed epithelial cell is not. They are the oxidoreductase. Oxidoreductase is upregulated by autocrine osteopontin C. And these oxidoreductases increase the level of ATP in the cell and inhibit the anoikis, the programmed cell death that is caused by the adhesion. Well, it was actually a fit what we knew about anoikis. We know that cells, once they are detached, have a much higher energy requirement than cells that are not detached. So somehow the ATP in the cell needs to be boosted. If it's not boosted, then the cell dies from anoikis. This is what we've learned. Metastasis genes support anchorage independence in an autocrine fashion. And because of these studies, because of the somewhat initially surprising finding of oxidoreductases being upregulated, we had to ask, well, what is the metabolic basis of anchorage independence? What goes on inside these cells once they are detached from the substratum and just float around in the circulation? Right? The tumor cells that circulate in the bloodstream or in the lymph vessels for weeks before they actually penetrate into their target organ. Well, for starters, we double transfected. Our MCF7 cells are tumorigenic, non-invasive cells. They were actually derived from a patient sample. And well, because they are endogenously totally devoid of osteopontin, they are easily transfected with splice variants. So we had from our previous studies, our vector control, our single transfected osteopontin A expressing cells and C expressing cells. We made double transfectants, and we realized that actually each and every one of these bars is significantly different from the next lower one to its left. And so osteopontin A and osteopontin C can collaborate. They can synergize it. We don't know yet whether it's additive or synergistic, but it's certainly statistically significantly higher clone formation when you have the double transfectants in osteopontin A plus C as compared to osteopontin C alone. So we put these cells onto a medium that actually resembled the soft agar, that's polyhema, polyhydroxymethyl acrylate, is a coating for the cell culture plates that prevents the cells from adhering. So you can mimic anoikis with that system. You can easily extract the cells, and you do metabolomics of the osteopontin A transfected cells versus vector versus osteopontin C. Jump out and again, it was a little bit of a surprise. It was something we had not predicted. We were not expecting to find that with osteopontin a whopping upregulation of glucose. There was the prominent finding in the metabolic change that glucose was dramatically upregulated in the osteopontin A expressing cells. We had known from previous studies that we had done, right? We've been at this for a few years and we've tried various technical approaches. One of them was a protein DNA array that looks at transcription factor binding. And we had known that STAT1 and STAT3 are upregulated by osteopontin A. So we took STAT3 and manipulated it in the osteopontin A expressing cells. On the top left, the graph shows the soft agar colony formation in cells that were treated either with a permeable STAT3 inhibitor peptide, so they are cell permeable peptides that inhibit STAT3 inside the cell, or a control peptide, or not at all. Right? So we have osteopontin A treated with the STAT3 inhibitor peptide, and the soft agar colony formation goes down to a statistically significant extent. We treat osteopontin C transfectants in the same way, and they are resistant to the STAT3 inhibitor peptides. Same thing with vector. And we followed that up with a genetic approach, right? We transfected the cells with either constitutively active STAT3 or dominant negative STAT3. And we see that dominant negative STAT3 knocks down the osteopontin A-induced colony formation in soft agar, which doesn't affect the osteopontin C-induced colony formation at all. And in vector, the dominant negative does not affect the colony formation of the vector, but the constitutively active STAT3 does increase the clone size to a statistically significant extent. Right, so dominant negative STAT3 can interfere with the osteopontin-induced signal, and constitutively active STAT3 can mimic at least part of the osteopontin A effect. You see osteopontin here and a constitutively active STAT3, so that's only a part of the osteopontin A effect. It's not a complete mimicry. And what you see on the bottom here is what we expect from a metastasis gene. Right? You put these cells into regular plastic dishes and measure proliferation. And there is no change in proliferation. Whether STAT3 is there, is upregulated or downregulated. Right? This is a metastasis pathway. This is not a growth pathway that we're dealing with in terms of osteopontin A and STAT3. Right? Then we took these STAT3 cells and did our metabolomics, and we see that STAT3 actually is upstream of the metabolic changes. So the osteopontin A signal to change the metabolism of the cell goes through STAT3. All right, this was all intriguing. It was all somewhat surprising. The one thing that was nice about it was very clean. The data were very clean. Well, of course, we were committed to osteopontin C. We are committed to metastasis formation, and osteopontin C is a much more prominent player than osteopontin A in inducing it. And so we did our metabolomics on osteopontin C, and it was much, much more challenging to figure out what's going on. You see there is much more diversity in the metabolites. The upregulation is much less striking. Some of them 15-20% upregulated. They are statistically significant. And so after some work and going from microarray, metabolomics, searching databases, and trying to put all of the pieces together, we were able to find that actually all the upregulated enzymes or metabolites, and they are boxed in red, from all the various experiments that we did. As I said, from microarray, from metabolomics, from even searching public databases for osteopontin-induced gene upregulation. All these molecules that are boxed in red have evidence in favor of being upregulated. They belong to three converging pathways. You see, of course, on the left, the canonical hexose monophosphate shunt glycolysis, tricarboxylic acid cycle leading into the mitochondrial respiratory chain. Well, and so we infer from our data that osteopontin C does increase this pathway and lead to increased ATP generation. Now, a second pathway, and that's one that is actually not often mentioned in the literature when you talk about metabolism. There is a channel protein on the cell surface that can basically bypass and synergize with the glycolysis and TCA pathway. That would be aquaporin 3. Aquaporin 3 allows glycerol to go into the cells, activate glycerol 3 phosphate. So the glycerol phosphate shuttle is sort of a lateral pathway leading into the respiratory chain. But you see, this complex down here is the same as what we have here. So this pathway feeds downstream of DCA into the mitochondrial respiratory chain. By doing so, it also increases ATP. And so we have two pathways, two synergistic pathways leading to increased ATP generation via the mitochondria. And there's a third pathway, and the third pathway via glutamine and glutamate. Glutamine and glutamate can feed into serine synthesis. And we have an upregulation of two of the downstream targets of serine, and that is glycine and creatine. Creatine actually can regenerate utilized ATP. Right? ATP is utilized, it gets broken down to ADP and AMP, and creatine can help in the regeneration. And so we have now three converging pathways, all focusing on ATP. We have now pathways that are canonical, excess monophosphate shunt glycolysis, TCA cycle. Pathways that actually are synergistic but only converge at the mitochondrial respiratory level, and that is our aquaporin glycerol phosphate shunt pathway. And then we have a third pathway that sort of circumvents the mitochondria and can regenerate utilized ATP via creatine. And actually, if I may call this to your attention, the previous slide that showed the upregulation of phosphoethanolamine and phosphocholine, we believe is not functionally important. We believe that this is a reflection of all the serine being funneled into ATP generation, and therefore upstream these two metabolites building up. And actually, it's not shown on the slide. It's in the paper that we believe that phosphoethanolamine and phosphocholine are actually backed up in the metabolic pathway because there's not enough serine to convert them further. And of course, we directly measure the ATP levels, and you see that the ATP generation increases as we increase our soft agar colony formation. The osteopontin A has a small effect, osteopontin C has a bigger effect, and when you combine them, you have a very dramatic effect on the increase of ATP levels inside the cells. And it's all statistically significant. So this is our most recent insight that we've gained into how cells are able to form metastasis, and this is a focus on anchorage independence. We realize that osteopontin A stimulates the generation of glucose. The glucose is then utilized by osteopontin C in order to feed through the glycolysis and the mitochondrial respiratory chain and generate energy. So osteopontin C generates ATP, which is facilitated by osteopontin A, which upregulates the glucose in the cells. All right, so we've been working on this molecule for a very long time, and there are a few insights that we believe we can state with confidence. We believe that for metastasis, just like for all the other aspects of cancer, there are specific genes that underlie the phenomenon. So there is a genetic basis for metastasis formation. Metastasis genes are stress response genes. These stress response genes in cancer are aberrantly expressed or spliced. They are not mutated. And when there are mutations, the mutations are in the regulatory regions of these genes, not in the coding regions. And metastasis genes support anchorage independence in an autocrine fashion. Now, very recently, our work has indicated that anchorage independent survival and expansion are achieved through metabolic adjustment within the de-adherent cells. And we have an idea of how that works. There are distinct metabolic changes that are induced by osteopontin A or osteopontin C, the two splice variants. And I should probably mention that C is never expressed without A. Right? The wild type form is always there, because the RNA for the wild type form is made first, and then it is spliced to generate osteopontin C. We now know that glucose uptake is an essential part of the anchorage-independent survival, and that is induced by osteopontin A, and that glucose is utilized by osteopontin C in order to satisfy the energy requirement by cancer cells in the circulation. Oopsie. Okay, yeah, so with that, we've tried to gain additional, I tried that one there. So with that, we've tried to gain additional insight into the molecular mechanisms of cancer. The goal, of course, being that we find new drug targets for the one aspect in cancer that is undertreated, and that is metastasis. Thank you. I think we do have time for a few questions. Yes, please.
I ask you about the splicing. Do you believe that, the splicing arrangement, does that make all the difference for the cells that you're looking at?
For all we know, yes. Splicing occurs only in chimpanzees and humans, not in mice. Mice have been used for metastasized studies, but these metastases don't form at the same abundance as in humans. And I believe that actually osteopontin A is a very poor metastasis generator, and osteopontin C is a very powerful metastasis gene. So I believe in part the paucity of modeling human cancer in mice is generated by the absence of osteopontin C.
Okay, thank you.
I wonder, because in your data you show that lung meta and liver metastasis is related to osteopontin protein. So we know that brain metastasis is different story. So is osteopontin C also related to brain metastasis because we that need to pass BBB?
Is osteopontin C also related to brain metastases? Actually, there are right now close to 20 papers in the literature focusing on osteopontin splicing in various cancers. There are, to my knowledge, 2 of them that have associated osteopontin C with brain metastasis. Actually, one of them with primary brain tumors, the other one with secondary brain tumors. So two that have associated osteopontin C with brain tumors, one of them is primary brain cancers and the other one metastases. So yes, I do believe that it is important for those 30 types of cancer that make osteopontin splice variants, that the splicing is also important for their brain metastases.
Beautiful talk. Given the importance, anybody tried to manipulate this pathway for therapeutic purposes?
Has anybody tried
to basically target osteopontin for therapeutic purposes?
Yes. The problem with osteopontin, the pan-osteopontin, is twofold. Number one, it's a very powerful Th1 inducer cytokine. So when you knock it out, you get substantial immune deficiency. And number two, the protein is highly modified post-translationally and post-transcriptionally, making it difficult to find the right domain for a drug target. The reason we formed a biotech company around osteopontin C was, of course, the attempt to generate an inhibitor. We have struggled with a splice variant-specific neutralizing antibody. We have a polyclonal, and they have been able to make it reproducibly. The domain around the splice junction is not predicted to be very immunogenic. We've gone through several attempts of making a monoclonal that have not yielded a specific one. The monoclonals that we have cross-react with other acidic domains. And the problem is when you have the splice junction and you make an antibody to it, you shift a little bit upstream or downstream, you get cross-reactivity. Right? You're tied down to just about 10 amino acids around the splice junction. So it's been very, very challenging. We haven't given up, but we did not anticipate the level of difficulty that we have encountered. Whenever that happens, I'm very optimistic it will be a very useful agent clinically.
But also the fact that this is clearly a pathway as active in normal physiological conditions, right? If you target it, do you see any off-target effects? Would you expect any?
See, with osteopontin C being cancer specific, I'm very optimistic that we don't see off-target effects. The whole excitement about metabolomics, as much as it has yielded valuable insight, sort of negates a major part of cancer research, That has been the accomplishment from 1976 with the discovery of the first endogenous oncogene, and then the 1980s with the signal transduction. This research has given us very specific drug targets. Right? BCR-ABL is a mutated kinase. It's a very specific drug target in CML, distinct from Abelson kinase in the healthy cell. The mutated EGF receptor in lung cancer is a very specific, qualitatively different drug target in the cancer from the normal cell. So molecular biology from the 1970s and 1980s onward has given us drug targets that are qualitatively different in the tumor versus the normal cell. Some of the research that we all have been doing, and of course I also am in the metabolomics area like everybody else who wants to be fashionable, but we are not paying enough tribute to the circumstance that those pathways, as you just pointed out, are very common. We're now going back to quantitative differences between the tumor and the normal cell. So this is going to be a limitation in terms of the clinical translation of that work. So we need to focus on drugs that have targets that are qualitatively different between the cancer cell and the normal cell.
But so osteopoietin C, as you said, is cancer specific, right?
Yes.
So I guess my question is that could that be a potential tumor-associated antigen? Because now the field is kind of burgeoning with excitement. Now we don't really have a good tumor-associated antigen to be used as a target of vaccines. Has anybody tried this? I mean, is it
I need to talk to you in the coffee break about some of the research that we are doing. It's a secreted molecule, right? What portion of that is presented in the MHC and in which one, we don't know. An anti-osteopontin C immune response, I'll talk to you about ongoing research. Maybe I can take one more question if you forgive me, and then we really have to move on, please.
Splice variants tend, as hard as we think, to be very specific. They tend not to be specific but transient. Have you looked for osteopontin C during development?
Well, actually not, no. We're talking about the adult healthy organism.
My guess is
there's a possibility.
Yeah,
yes.
It might not be, because the field has been burnt before thinking that there are TSTAs.
Yes. And
other than translocations.
It could very well be, but it would still make it specific in the adult healthy organism, right? It would be absent. But yes, the developmental aspect we're not set up to do. We haven't found the right collaborator. I do agree with you. There's a significant possibility that we would find osteopontin C right there. Okay, thank you very much.